My name is Oscar Palomares and I am professor at Complutense University of Madrid. It is my great pleasure to share with you today our last findings published in Allergy, showing that Tezepelumab reversed TSLP-driven immunometabolic rewiring in human type 2 conventional dendritic cells, thus restoring proper adaptive immune responses. TSLP is an epithelial alarmin that plays a very important role in the regulation of inflammatory responses in asthma and CRS with nasal polyposis. TSLP acts on a plethora of immune system and non-hematopoietic cells that express the functional receptor for this alarmin. Tezepelumab is a human monoclonal IgG2 antibody that blocks TSLP and avoids its binding to the functional receptor, thus impairing downstream signaling functional properties. Tezepelumab has demonstrated safety and clinical efficacy in asthma and CRS with nasal polyps. However, the mode of action is not yet completely understood. Especially the role of TSLP on dendritic cells is not fully elucidated. Therefore, the aim of this study was to determine whether TSLP induces a pathogenic immunometabolic program in human type 2 conventional dendritic cells and whether Tezepelumab can directly reverse this process. For that, we purify type 2 conventional dendritic cells from peripheral blood of non-atopic healthy donors and stimulated these cells either with TSLP alone or in the presence of Tezepelumab to analyze the metabolic reprogramming using Seahorse technology. Glycolytic rate and cell mitostress assays clearly demonstrated that type 2 conventional dendritic cells treated with TSLP displayed a significantly higher basal and compensatory glycolysis, as well as significantly higher basal and maximal respiration and ATP production coupled to respiration when compared to conventional type 2 dendritic cells just stimulated in the medium. Importantly, Tezepelumab in the orange bars were able to significantly reduce all this metabolic hyperactivation induced by TSLP, thus demonstrating that Tezepelumab effectively blunted the TSLP-induced metabolic rewiring in human type 2 conventional dendritic cells. Next, we wanted to study how type 2 conventional dendritic cells could polarize T-cell responses when stimulated with TSLP alone or in the presence of Tezepelumab. And for that, we performed the co-culture experiments that you can see here in this slide. T-cells generated by type 2 conventional dendritic cells treated with TSLP produced significantly higher levels of IL-5, IL-13, TNF-alpha, and lower levels of IL-10. Thus suggesting the generation of pathogenic Th2 cells. Tezepelumab was able to inhibit the induction of type 2 cytokines, pro-inflammatory cytokine TNF-alpha, and to restore the production of IL-10. Interestingly, our correlation analysis demonstrated that the TSLP-induced metabolic hyperactivation of conventional type 2 dendritic cells was significantly and positively associated with the number of pathogenic Th2 cells generated by these dendritic cells. Human type 2 conventional dendritic cells stimulated with TSLP produced significantly larger numbers of FOXP3 positive regulatory T cells, which was significantly reduced by Tezepelumab treatment. Interestingly, the suppressive capacity of the regulatory T cells generated in the presence of TSLP was significantly lower than those observed in regulatory T cells generated under the control conditions, and this was again restored by Tezepelumab treatment. Correlation analysis showed that TSLP-induced metabolic hyperactivation of type 2 conventional dendritic cells was inversely correlated with the functional activity of the regulatory T cells generated by these human type 2 conventional dendritic cells. Collectively, we showed that Tezepelumab reversed the capacity of TSLP-activated human type 2 conventional dendritic cells to polarize pathogenic T-cell responses by mechanisms that depend on the restoration of the immunometabolic rewiring induced by TSLP. So in summary, these results provide novel mechanistic insights into how Tezepelumab might reshape pathogenic adaptive immunoresponses. This data supports also that upstream epithelial alarmin blockage is a strategy to interfere with early disease-driving immunoprograms in type 2 inflammatory airways diseases. And that is all I wanted to share with you today. Thank you very much for your attention.